please.
All right, thanks, Dennis, for the invitation. I had a full Proustian moment when I entered this room because I think it was in 2011, and I visited Maria, my wife, when she was doing a postdoc in Cambridge. I entered this room and I saw, I think it was Daniel Schechter giving a talk in the St. Will Club. And, now I'm here, so, getting a little bit anxious here, but okay. So what I would like to do for the next 45 minutes is to revisit two classical theories which have, centrally shaped memory research. One is the famous fire together, wire together idea put forward by Donald Hebb some 70 years ago. And the other one is hippocampal indexing, which was proposed some 40 years ago by Taylor and Shannon. And so those would be the first part about Hebbian learning, the second part about hippocampal indexing. And at the end, I would like to show how research that is driven by these ideas, its fundamental research, can actually lead to very specific and very actionable plans for neurotechnology to help patients with memory problems. And this talk will also, to some extent, tell a personal story as to how I developed from a basic neuroscientist being purely driven by curiosity to a somewhat more goal-oriented scientist who actively seeks to translate the findings that he finds in the lab to benefit society. And ultimately society, that's the funding body we report to. Okay, right. So as a result of that, I am now also advisor to companies like Clarity, who do a fantastic job in translating findings from me and others to develop wearables for patients with dementia. And so now I have to disclose this in my talks. So that's the disclaimer. All right. So let's start with that story. About 6 years ago, I was at the conference, and I, after a whole day of sciencing, I retreated to the bar to have a drink. And I met this guy, that's Bishan Pesaran, and he asked me this question, said, "Simon, what's your 20 years research vision?" And my reaction was that. So I was embarrassed because I didn't have any idea how to answer the question. If, at the push, you could cobble together some ideas and then maybe come up with like a 5 years plan, but that was it. And so that question, right, the 20 years research question, this was one of these career-changing moments. They tend to happen at bars often. And I will give you the answer to the question, but to get the answer, you have to hang about till the end of the talk, I'm afraid. What this is it's an example of how you maybe should have a long-term vision for your research, but also it's an example of an episodic memory, right? So this event happened 6 years ago at a specific location, at a specific time. It was in Chambéry at a conference. And it's an example of an episodic memory, right? So it has a unique temporal spatial reference point. And those memories only happen once, right? You only get one first day at school, you get one first kiss, and so on and so forth. So that's an example of an episodic memory. And the question that we ultimately want to answer is how can our brain form these memories? How can they hold a record of such fleeting experiences? And one answer that was given 70 years ago, and you all know that, of course, was by Donald Hebb, who suggested that it is the firing patterns of neurons, much like as we walk in snow, leave footsteps that allow us to trace back where we came from. These firing patterns leave traces in those connections, which we call synapses, and those connections will change as a function of these firing patterns, which coined the famous, almost a cliche now, neurons that fire together, wire together. And we know from a lot of studies that have been done in animals that there is such a principle indeed that is realized in the brain. Some 50 years after Hebb was proposing this idea of wire together, a study in a slice preparation. And I'm very happy that Ole Paulsen is here. He knows this much better than I am, than I do. Found a principle called spike timing dependent plasticity, which very much behaved like, or what was basically the Hebbian learning principle embodied in the brain. So spike time independent plasticity, what it suggests is that if you have a presynaptic neuron shown here in green that projects to another neuron shown in purple, and that presynaptic neuron fires before the postsynaptic neuron fires, then you get long-term potentiation. And the closer in time this delay is, the shorter this delay is, the stronger will be the increase in that connection. If this firing pattern happens the other way around, so the postsynaptic neuron now firing before the presynaptic neuron, you get long-term depression, that is a weakening of that connection. And again, the timing is emphasized here by this exponential function. So timing really matters, and the sequence matters. And this is why gamma oscillations have been suggested to play a crucial role in there, because gamma oscillations have a frequency of about 40 to 80 hertz, so a cycle length of about 25 to 12.5 milliseconds. So those oscillations can, in principle, entrain neurons to exhibit these, temporarily fine-grained firing patterns. And it is, of course, a big jump to go from a slice preparation, of basically a dead brain, the slice is being kept alive, to a human actually forming memories. So the question that we asked is, can we find such a realization of the principle in a human that is actively forming a memory? And to this end, we turn to epilepsy patients to answer the question whether we can relate the co-firing of neurons to these oscillations. And so what you here see is an epilepsy patient who's getting implanted with an electrode like this by the neurosurgeon Ramesh Chavaraja at the Queen Elizabeth Hospital in Birmingham. And so this electrode has these tiny wires that stick out at the tip. And with these tiny wires, we can record signals like these. Of course, these electrodes go in purely for treatment reasons for the patient. They are to ascertain where the epilepsy is coming from, so that the neurosurgeon can then basically extract that brain region. But whilst the patient is in the hospital, they're often bored out of their mind because they sit there waiting for seizures to happen. They can do experiments. Okay, so with these microwires, we record signals like these, which is a complex signal, and you can see that you can see the spike here on top of like the background activity. So the background activity, that's basically all the cells in the surrounding that are talking to us. And this spike is a neuron that is really close by, which is also talking to us at this time. And we can filter the signal to separate the local field potential. So those would be like, your more or less rhythmic background activity from the spiking of these neurons. Now, all we need is to give our patient a task to do, and this was a memory task where we asked the patient to form associations of these three images. So they saw a picture of an animal, then the picture of a person in the place, and they had to come up with a story that would link these three images together. And then later on, in the acute recall task, we presented them with the picture of the animal, and they had to tell us how many pictures do they remember, and then confirm their answers. And what this allowed us to do is to look at what is happening at the time point of encoding that is predicting later memory success. And specifically, can we see something like higher synchronization or synchronization at tight time windows and co-firing of neurons during that time that will predict the later memory success, as would be predicted by spike time independent plasticity or Hebbian learning. So as a first step, we looked at spike field coupling. So spike field coupling is basically a measure where you take the spike and you correlate it with the local field potential with a frequency filtered version of the local field potential. And if you get high spike field coupling, means that neuron will always fire at a certain phase of that oscillation. If you get low coupling, that means the neuron's firing all over the place. It's not really, related to the phase of the background oscillation. You can do this in two ways. You can measure the local field potential, or the oscillation and the spiking with the same electrode, or you can measure the spike in one region and then measure the local field potential in another region. This is typically interpreted as a directional measure of coherence, because basically the spike of that neuron ends up being the postsynaptic potential in this other region. Now, when we looked at the local coupling, we found a strong gamma oscillation between like 60 and 65 hertz. And when we looked whether this oscillation would change as a function of memory, we found this. We found that later successfully remembered trials were showing a slightly faster frequency than later forgotten trials. So this is one example of one such spike LFP pair, and you can see the blue curve, that's the hits, shows a slightly faster frequency than the misses. And that was true on the population level. And so this result is consistent with the spike time independent plasticity idea, because you have a faster frequency, which means that the neurons are synchronized at tighter time windows. Now, we did the same analysis for the distal pairs, and here it showed it was theta oscillations were telling the interesting story there. So we found, very much like we found for the gamma oscillation, we found that fast oscillations at theta were indicative of a successful memory, and slow oscillation indicated a non-successful memory. And that was true at the population level as well. So again, you see this fast oscillation, predicted good memory outcome, and slow oscillations predicted a slower memory outcome. And so this allowed us to basically form pairs of neurons. So we have like a downstream neuron that is sending basically the theta spikes to some other neuron. And then there we have a local neuron which is listening to the gamma. And in an analysis I don't show you for time reasons, but just have to believe me, that's the case. We showed that the gamma oscillations were indeed coupled to the theta oscillations. The more they were coupled to that, the more successful the memory outcome was. So we thought, okay, what's the business of all that synchronization stuff, right? The business of that synchronization stuff is ultimately to allow an upstream neuron to transmit its information effectively to a downstream neuron. And if that's the case, then what we should see is if the downstream neuron fires a spike, then the upstream neuron fires a spike, the downstream neuron should also fire a spike as a result of that. And the closer in time that occurs, right, the closer this co-firing occurs, the more this should predict a successful memory outcome, because this follows from spike time independent plasticity, right? And so this is one example of one such neuron pair. And you can see that indeed we find faster co-firing for later successfully remembered trials versus later not remembered trials. And that's the result on the population level. So this does confirm one prediction of spike time independent plasticity. Co-firing at temporally tighter time windows, right? Co-firing at shorter latencies predicted successful memory outcome. That's nice, but the question is this correlation or is this causation, right? So in that case, we didn't really manipulate the synchronization, we just observed it and correlated it with memory outcome. And we thought very long and hard how we can get at correlation in humans. Of course, in an animal, you do some fancy optogenetics, but you can't really do that with humans. So what we came up with was this. I'm going to show you a video, which, if things go well, should also be accompanied by sound. And this is flickering, so if you have photosensitive epilepsy, then you probably should not look at this. Okay, so what you saw was a video that was flickering at a very fast rate, should have been 35.30 37.5 hertz if the refresh rate of the thing is 70 hertz, but it might not be. But the sound was also modulated at that same frequency, right? And we used that in an experiment where we asked participants to form memories of these video sound combinations. So the sound was not related to the video, right? It was an arbitrary association that the participant had to form, and both were modulated at this fast gamma frequency. And now this allowed us to place the video and the sound in different phase offsets with respect to each other. So we would have a condition, where both were perfectly synchronized, or we could, desynchronize the two streams such that the video would lead the sound by 6 milliseconds, so 90 degrees, or we have them both in antiphase, or we could shift them such that the sound would lead the video by 6 milliseconds. Or in other words, the sound would be lagging the video by 270 degrees phase offset. So following STDP, these are the interesting two conditions, right? Because here you have the video leading the sound by a short delay, and here you have the sound leading the video. So that's what happens at encoding. Now we need the behavioral readout of that, like the pre and the post-synaptic connection in the later test. So how do we do that? We did it this way. So we presented either the video and asked the participant to retrieve the sound, or we presented the sound and asked the participant to retrieve the video. So this would allow us to probe how we change these, one-way streets, if you will, from either the visual to the auditory domain, or from the auditory to the visual domain. And I can see some faces frowning here, like, how does this look like? We were the same. That's why we came up with this very simple computational model to simulate the results, so that we can have a clear prediction as to what should happen, what should we observe in behavior. And what we should observe in behavior, if spike time independent plasticity is a thing in episodic memory, is this: we should basically observe an increase in memory performance when we cue with the video for the condition where the video is leading the audio by a short delay. And we should see an increase in memory performance for the 270-degree condition where the audio is leading the visual by a short delay, okay? So that's the prediction. First, we needed to confirm that our stimulation worked, and this is the EEG results. So we socialized the visual and the auditory cortices and measured the phase delay between those two regions, and then basically sorted the trials based on which phase delay we got. And so those would be the two interesting phase spins. So this would be where the video leads the audio by a short delay, and this is where the audio leads the video by a short delay. And these are the behavioral results, which confirmed indeed what our computational model predicted. So these, the memory performance was better for the visual condition when we cued with the video and asked participants to retrieve the sound, when the video was leading the sound by a short delay, and the other way around for the condition where we cued with the sound and asked participants to retrieve the video. So in this, so when we published this, a reviewer asked us to also do some source localization to see whether we can observe some effect in the hippocampus. Had very little hope for that, but gotta do what the reviewer says, right? And to my surprise, we found a pretty interesting effect in the hippocampus. So we found that the 37.5 hertz, so that's the signal, right, that these movies were flickering at, was stronger if the trial was later remembered than when it was forgotten. And that's I think a pretty interesting result, because it suggests that the level to which the hippocampus gets entrained by these external stimulus depends on memory, probably because it happens at encoding depends at attention, which we know is a major gatekeeper to the hippocampus. And I think that's fairly interesting, because there are some studies in that space that question whether the hippocampus gets entrained by these 40 hertz oscillations. It's a point I may come back to later. this is data that's coming fresh from the oven, so to speak. Victor literally yesterday sent me the results. So we did another study where we tried to replicate these results, but not with flickering videos. They were flickering themselves on the screen, but with so-called shutter glasses. So these are glasses that James Dorset and Sterling developed. These are glasses which will basically go from dark to opaque. Sorry, from opaque to transparent by a voltage change, and this voltage change can be triggered with an Arduino device. So in other words, these glasses can be made to flicker at 40 hertz. You get brilliant timing. It's so accurate. It's amazing. And we did the same experiment. So now with people just wearing these glasses, and the video was not itself luminance modulated. The glasses were providing the luminance modulation, and we had the same sounds, and we had these two conditions where basically the video was leading the sound, or the sound was leading the video, and confirmed with our EEG analysis that this actually worked somehow. And we could indeed replicate this result, that when we cued with the video and asked people to retrieve the sound, we get the better memory performance for the 90 as compared to the 270 degrees. And I find this really, really interesting because these shutter glasses, they're really cheap. You can basically produce them at 50 quid or something. So this could be something that people could wear actually as they go about their daily lives, and it could be something that we could use to translate these findings to improve memory, for instance, for Alzheimer's patients. And Eleonora Marcantoni at the moment is working on a project where we try to adjust the frequency at which we flicker to the momentary frequency that the hippocampus has at this moment in time. We do this with MEG, where we source localize the hippocampal activity, extract the frequency, and then flicker with that exact frequency that the hippocampus has in this moment. And as I said in the beginning, basic scientist driven by curiosity, but now wants to translate some of the stuff back to society. So that flicker business is especially that 40 hertz flicker stuff, that could have a real impact for patients with Alzheimer's, because studies by Li Hui Tsai's lab at MIT have shown that if you flicker a mouse model with Alzheimer's at 40 hertz, it actually reduces the amyloid burden and improves memory performance in these mice. And it doesn't do so, it's not a magic thing that happens. It triggers a lymphatic response. And in that paper, they spell this out, whereby basically the neurotoxins are flushed out of the brain and then degraded in the lymph nodes. So that 40 hertz stuff could actually have some real life impact. And you all know probably Alzheimer cases will triple, right, in the next 25 years. And we have nothing, right? We have no treatment option. Most of these cases will come from low economic income countries. They would have not access to terribly expensive medicine. But, a flickering light source, that could be something that could be used, that could be useful for these. So to sum up the first part, what I've shown you is the synchronization of neurofiring to gamma and theta oscillations correlates with memory, and it correlates with co-firing of neurons. And when we modulate synchronization at the gamma frequency via multisensory stimulation, which I also call the poor man's or poor woman's optogenetics, right? We actually get results that are in line with Hebbian learning or spike time dependent plasticity. And I think this is really cool because it opens up a new avenue to investigate time-independent plasticity mechanisms in humans. It allows us to build a bridge between basic, web neuroscience, slice preparations, to humans actually, doing stuff, memorizing things. And potentially this could deliver a scalable treatment option for Alzheimer's disease. Okay, now to the second part, the memory indexing part. So Taylor and Deschena put forward this idea that what the hippocampus does in order to allow us to hold a record of these complex memories, you know, like me meeting Vijan Pesaran at the bar 6 years ago and so on. So what the hippocampus does is it actually doesn't store all the details that relate to that event. But what the hippocampus stores is like a very abstract, an index, a pointer that will point to all the different elements. And we have the neocortex, which analyzes, you know, everything we perceive in great detail. So the neocortex would hold all that information, and the hippocampus just basically represents a pointer which acts as a glue to hold together all these different things. So to this degree, the hippocampus would very much act like a librarian, or like, you know, a very sophisticated search engine. You type in some keywords, and then you get the books that would, or the book that you're looking for. And the librarian might not have read the book, might not know what's in the book, but they know where you can find the book. Okay, so that's in a nutshell. That's the indexing idea. However, if we look at, you know, which neurons are observed in the human hippocampus, then probably the most dominant type of neuron that have been found are concept cells, which do not behave like index cells. So these are neurons that my dear and very critical friend Rodrigo Quiroga 20 years ago, right, has found. So these are these Jennifer Aniston neurons. So these are neurons that will fire to a certain person, a certain location, or a certain concept. Could also be a mathematical equation, if you're really into math. And these are just two examples of concept neurons that we observed. So where the patient really was into the rock singer. Does anyone know who that is? Pink. Nice one. Yeah. So these days, no one knows anyone who Pink is. We have to find a Taylor Swift neuron, I think. So that neuron, whenever a Pink song was played or presented an image of Pink, then that neuron would fire. And here's another neuron for this, Sydney Opera House. And these neurons are found with so-called visual tuning tasks. You present a lot of images and repeat every image 7 times, and then you find the neurons that would fire to a specific image. And these neurons, the important bit is that these neurons will fire whenever something is happening that makes reference to the concept. Okay, so they have no respect for context. Context doesn't matter, right? So they are really a hard-coded thing in the hippocampus that always goes off when something is happening that makes a reference to the concept. So per definition, they cannot code for a unique experience involving that concept, okay? Now, if we take, what we want to explain, though, is how can we remember an event that involves multiple elements, like you meeting a friend for coffee, right? How does that happen? And Rodrigo Quiroga says, well, it's all done by concept neurons. So you have a concept neuron that is maybe firing to Miles Davis because there's a Miles Davis song playing in the background somewhere. I'm into Miles Davis. So. And another concept neuron for your friend, and another concept neuron maybe for the waiter, and then together these neurons cover this event. But I was always dissatisfied with that theory because our lives are, they are a bit samey, right? So we go to the same locations, meet the same people. So if we only have concept neurons, I think we really have a difficult time teasing apart those very, very overlapping events. And what we were wondering about, can we find so-called neurons that would behave like a hippocampal index neuron would, where a neuron would basically code for a conjunction of all these elements together. And that was the question that Luca Colibius set out in his PhD, which he pursued. Started, I think, 5 years ago. Jesus, time flies, huh? So, so what did Luca do? So Luca ran the same experiment as we did before, with these images of animals and people and places, and the patient had to form a memory of these. And then what he analyzed was the spiking of the neurons during the encoding time and during retrieval. And what he was looking for was particularly neurons which would increase their firing rate for specific events during encoding and retrieval. So here on the x-axis, would be the event that was encoded, and you can see in purple the firing of that hypothetical neuron during encoding, and in orange, the firing of the neuron during retrieval. And he calculated the element-wise product, so the combined firing rates during these both events, and then did some shuffling to see whether he finds a neuron that sticks out for a singular event, which would increase its firing rate during encoding and retrieval for particularly this event, but not other events. And he found neurons like this. So that's a neuron which is, not firing a lot. I mean, we recorded this neuron for like an hour or so, and it fires 1,000 spikes. That's not a lot. That's a firing rate of 0.5 hertz or something. And this neuron increased its firing rate vigorously during encoding and retrieval of this particular event, and maybe also a little for this event, but it wasn't interested in any of the other events. And we labeled this neuron episode-specific neuron, right? Because it would only increase its firing rate during encoding and retrieval of a particular event, maybe two. And we found a higher number than we would expect by chance. And now you could say, well, could be an episode-specific neuron, could be an indexing neuron, right? But could also be a concept neuron. Because who's to say that this neuron doesn't just fire for one of these elements? So say this, it's a tiger neuron, and it fires during encoding when you present the tiger, and it then also fires during retrieval when you think of the tiger, which was something that Gelbart-Sagiv observed. So we did the experiment again, but now we screened for concept neurons and excluded those concept neurons from the analysis. And we replicated the initial finding. We again found a bigger than chance number of neurons, which would increase the firing rate specifically for events during encoding, for particular events during encoding and retrieval. There's a couple of more results and controls that we did. So we also looked whether we could find these neurons outside the hippocampus. We also collected enough neurons in the parahippocampus that would allow us to do this analysis. We did not find episode-specific neurons in the parahippocampus. These episode-specific neurons seem to be limited only when the episode is successfully encoded and retrieved. We did not find them if the memory was not successfully formed. Based on their wave shapes, these episode-specific neurons seem to be primary neurons. The wave shape is a little bit wider and the firing rate is a little bit lower than for the rest of the neurons. And interestingly, these neurons don't only have a rate code, so they just not only increase the spikes, but they also have a temporal code. So the time delays between the spikes also correlates above chance between encoding and retrieval. One frustrating bit is if you work with human single neurons is that we don't know where in the hippocampus they are. So we have no idea in which subfield these neurons live. And therefore we turn to high resolution 7 Tesla imaging to ask whether these episode-specific codes or concept codes would map to different subfields. And the hypothesis for this experiment came from a beautiful computational model was put forward by Anna Shapiro, where she suggested that maybe in the hippocampus we have a complementary learning scheme. You might be familiar with the complementary learning idea. You have the hippocampus, the fast learner that stores specific indices for unique events, and then you have all your semantic memory in the neocortex. And she suggests maybe you have a similar division of labor between hippocampal subfields, mainly between the monosynaptic pathway, which connects the entorhinal cortex to CR1, which according to her framework behaves much more like cortex and should do statistical learning and do more things that we would liken to be semantic. And then you have the trisynaptic pathway, which connects entorhinal cortex to CR1 via dentate gyrus and CR3 to CR1. Which which has a higher plasticity rate, so you get much stronger changes in the synapses in single trials, and also it's very sparse, and it should there's pattern separation and all these things, which sets it up to basically spit out episode-specific codes. So the hypothesis is that we should find concept cells in CR1 and episode-specific cells in CR3. Now, with 7 Tesla fMRI, we can't measure single cells. We get populations. So we need to do a little bit of simulation of our hypotheses to see, what would we expect by such population level measures. And so here's the model. Here's our thinking. So let's assume we have concept cells in CR1, and we ask a participant to to form memories of Brad Pitt, Taj Mahal, another version of Brad Pitt, the Eiffel Tower. Here you have an Eiffel Tower, and that's Nina Simone. And if you have concept neurons that will code for Brad Pitt, then these neurons should fire regardless of which version of Brad Pitt you see. And if you have neurons that code for the Eiffel Tower, these same neurons should also fire regardless of which version of the Eiffel Tower you show. In CR3, you should find a different level of coding. In CR3, you basically should see a unique population of cells that gets activated for each event, and they don't care about the overlap, right? They don't care whether you repeat the Eiffel Tower or Brad Pitt, and so on. And so, if we assume you have a population signal, then and we correlate the similarity of those different voxels of the different populations of neurons, then what we would expect is that for CR1 we should see a linear decrease in that similarity. So you should get, of course, the highest similarity between encoding and retrieval for repetitions of identical events. It should drop a little bit, but still be above chance for if you only have a 50% overlap, and then it should basically go down to 0. And this should not happen for CR3, right? For CR3, you should only see an above chance similarity between encoding and retrieval. If you encode and retrieve the same event, it should drop down right to baseline for 50%, and it should stay there. Okay, so that's the basic idea. And these are the results from that study. So for CR1, if you squint a little, you might say, it's a decrease, but it's not really, there's not really much there. But for CR3, actually, we did find a significant effect, and we did find above chance similarity for the same event, encoding and retrieval of the same event, and it dropped down to baseline for the 50% overlap condition. For the dentate gyrus, again, not much there. So this result confirmed this, event-specific coding for CR3. Now, what about concept cell-like signals? So in this experiment, we also repeated images. So it allowed us to extract the similarity between different versions of repeated stimuli. So here finally you have Taylor Swift, right? I have a 10-year-old daughter, so gotta have Taylor Swift in my presentation. And the idea is you have some voxels which sample a low number, just by chance, a low number of concept cells, and you have other voxels which sample a high number of concept cells, which would code for, let's say, Taylor Swift. And then you can correlate these two signals, these two vectors of voxels, and the assumption would be is you should, for repetitions of the same concept, you should get a higher similarity than for repetitions of different concepts. Okay. And this is the result of that analysis. So much to our surprise, we did not find concept codes in CR1. That thing was just sitting there very stubbornly at 0. But we did find concept cells, or concept cell-like signal, if you will, a concept cell population signal in CR3. So that did not confirm our hypothesis. There is a paper which it's not out there, it's a preprint, but it does suggest that at least in rodents, you could also have like more semantic codes and more episode-specific codes in CR3, and where you find those depends on how proximal or distal they sit with respect to dentate gyrus. So that could be consistent with that. Anyways, not everything works out the way you expect, right? That's the fun of doing science. So how can we reconcile these episode-specific cells and concept cells? How do they link. And for this, we took inspiration from Sheena Joslin's work, who has done fantastic work in rodents, and it gave rise to the engram allocation idea. So what did they do? So they measured neurons and rendered some of these neurons more excitable than the other neurons, either via genetic manipulations or via optogenetic stimulation. And this simple manipulation, just making some neurons more excitable, also seemed to allocate these neurons to code for an experience that the animal was experiencing at that moment. And then if you stimulate those neurons, then the animal would express a behavior that was consistent with that experience. So as if they would be just randomly grabbing a bunch of neurons and say, you code for that thing, and then we stimulate those neurons, and then the memory was being expressed. And that was, we took that as inspiration to come up with the idea maybe as to how these episode-specific neurons might get allocated in the first place, and then how some of these neurons might go on and be concept cells. So the idea is that some neurons at a certain time are just more excitable than the other neurons. They just happen to be because you have pockets of excitation that randomly wander around in the hippocampus. So those neurons should then get allocated to whatever happens in the neocortex, say an event where you meet your friend for coffee in a cafe. And then after some time, this excitability normalizes, and then you think about that memory, and then the memory gets reactivated. Because these neurons are then temporarily more excitable, you can change them. You can integrate this memory. And now if you think about, you first go and have coffee with this person you just met, and then you meet in a park, and you meet in a cinema and so on, and you constantly, think about, well, not constantly, but you happen to think about that person. And then you have many different reactivations of these episodic neurons, and some of those neurons will always get reactivated, right? So, and these neurons could then basically be the ones that then code for this shared element. So via a constant reactivation of a shared subpopulation of neurons, you can basically carve out concept neurons from episode-specific neurons. And so the provocative idea that we put forward in this paper is that every neuron starts first as just being, a bit more excitable than the rest of the neurons. Something happens, they code for that specific episode. And then some of these neurons will go on and develop into concept neurons. And this is consistent with the idea that we find actually more episode-specific neurons than we find concept neurons. So between 8 to 10% of measured neurons in the hippocampus will be concept neurons, and between 20 and 25% of the neurons we find to be episode-specific neurons. So that is consistent with that idea. We also did some sparsity analysis on the fMRI data that I don't have time to go into, but we can come back in the discussion to that. That also confirms this prediction, namely that concept signals are really sparse, episode-specific signals are not. Okay, right. So that's the conclusion of the second part. So we find episode-specific neurons in the human hippocampus, and they fire during encoding and retrieval of complex episodes. And that's consistent with this indexing idea of Taylor and Shanna. Episode-specific codes are found in the hippocampal subfield CA3, but we did not find them in CA1 at antigyrus. Concept codes are also found in subfield CA3. We have to wrap our head around that one. Concept codes are sparse. Episodic codes are not. That's the data I didn't show you. Just have to take my word for it, or ask me in the discussion. I can show you that. And episode-specific neurons may first be allocated to an event based on excitation, and then repeated reactivation of a subpopulation of these cells may transform some of them to be concept neurons. All right, so let's go back to that bar. And you may be wondering, so Simon, what's your 20 years research vision? So that question, you know, it was like, it really took me a time to figure that out. Like every morning I woke up, that question would be sitting there like an uninvited guest looking at me, what's a 20 years research? And it took me a while. But one day I woke up and I came up with an answer. And the answer is that I would like to build a prosthesis for memory, right? Bit of a crazy idea, but I got 20 years time, you know? And so the moment I formulated that research question, everything changed for me, right? Because from that moment on, when I, you know, as you navigate the muddy waters of academia, right? That is, you tend to get lost in stuff. And that thing just acted as my North Star. So, you know, whenever I did something, like, you know, like John Duncan here, one of your talks, you see, I want to go to Malaysia. What do I do with my right index finger, right? When you click on the mouse. And you're one talk I remember, anyways. So that's the thing, right? So when I'm doing something, I can ask, does what I'm doing, does that bring me towards that goal? So this was really an important moment for me, career-changing moment, I would say. All right, so we're building this prosthesis for memory. We're making baby steps, and this is one such baby step. So here we stimulated in the hippocampus with a rather big electrical stimulus, with 8 to 7 milliamps, so big stimulus. And it drove neurons, right? So this is one neuron that fired like crazy right after stimulation, but then it wouldn't fire a single spike. So that's probably not a good way to start, right? It's a bit like you're playing the piano with, you know, 2 big hammers. And what we're now trying to do is to be a bit more subtle and stimulate with microwire electrodes. And this is one simulation from a pretty cool toolbox where you can simulate what, you know, a certain stimulus would recruit in terms of neurons. And you can see how you can, you know, nicely recruit different populations of neurons with subtle changes, for instance, in the amplitude. These are not milliamps, these are microamps. So that's something we try to explore and then use this in patients, which are implanted with these electrodes so that we can allocate neurons, so make them just a bit more excitable than the other neurons during the encoding of a specific event. And then later at retrieval, stimulate those same neurons to see whether that triggers the reactivation of that event. And, you know, if that works, then we bring yourself into. All right, so I would like to thank you for your attention and the many collaborators that all, you know, contributed to the studies. Yeah, I guess it's question time.